Here is a micrograph of skeletal muscle. You can easily note its striated appearance. The striations are a result of unique anatomical arrangement of smaller microfilaments that comprise the myofibrils. As previously stated, these striations are composed of thin I bands that represent the location of the protein microfilament called actin, and thicker A bands that represent the location of the protein microfilament called myosin. The actin molecules are attached at one end to a filament structure that is made up of several different proteins known as the Z-disc. The space from one Z-disc to another is called a sarcomere and represents the basic unit of muscle contraction, meaning that muscle contraction occurs because of what happens within these regions. A sarcomere is about 2 micrometers in length. Therefore, one single muscle cell potentially contains thousands of sarcomeres. So now let's look a little more closely at the anatomy of the sarcomere. As you note, the sarcomere is made up of a repeating pattern of thick and thin filaments. Upon closer inspection, we can see how the thick myosin and thin actin filaments are arranged within the sarcomere. Once again, the thick filaments are composed of a protein called myosin. A molecule of myosin has a rod-like tail terminating in 2 globular heads. The heads are called cross bridges and link the myosin to actin during muscle contraction. Each thick filament contains about 200 myosin molecules, so that the filament is studded with cross bridges. The cross bridges are also specialized to contain binding sites for ATP, the source of cellular chemical energy. The I-band region contains the thin filaments that are composed mainly of the protein actin. Actin molecules have active sites that the myosin cross bridges attach to during muscle contraction. Actin appears as a helical structure that looks like a twisted double strand of pearls. 2 strands of a rod-shaped protein called tropomyosin spiral around the actin molecule and help to stiffen the molecule. In relaxed muscle, tropomyosin blocks the actin active sites so that the myosin heads cannot bind to the thin filament. Another protein called troponin binds to actin, tropomyosin, and ionic calcium. Both troponin and tropomyosin serve to regulate contraction by assisting in controlling the actin-myosin interaction. Okay, so let's begin to examine the physiology of muscle contraction. As you recall, the plasma membrane is a barrier to simple diffusion. The selectively permeable nature of the plasma membrane allows for the development of distinct and different intra- and extracellular environments. As a result of differences of ionic concentrations, the overall charge on one side of a plasma membrane may be different from the other. When this occurs, we say that the plasma membrane is polarized. Such is the case of the sarcolemma. Sodium ions present at higher concentrations outside of the muscle cell as compared to intracellularly. This is because at rest, the muscle cell is relatively impermeable to sodium. Sodium ions are unable to diffuse into the muscle cell. Potassium ions present with a higher concentration within the cell. However, also at rest, the cell is impermeable to potassium, so potassium remains in the cell rather than diffusing out. We therefore note that the outside of the membrane is more positive and the inside of the membrane is more negative. So at rest, the muscle cell is said to be polarized. This is important to understand because muscle contraction is a depolarizing event. As previously stated, skeletal muscle is finely controlled by the nervous system. The basic unit of the nervous system is the neuron. The neuron is a cell made up of three basic functional regions: the receptive region, which receives messages from other structures; the transmissive region, which physiologically transports that message along the length of the cell; and the secretory region, which transmits the message to another structure. In the case of a motor neuron, that structure is a muscle. As previously stated, each individual muscle fiber is innervated by a single nerve cell. However, a single nerve cell may control or innervate a few or many muscle cells. A single neuron and the muscle cells it innervates is called a motor unit. Although the secretory end of the nerve is very close to the muscle, the two structures do not touch each other. The space between the nerve and the muscle fiber is called the synaptic cleft. When calcium ions enter the secretory portion of the nerve, it triggers a release of an organic molecule called acetylcholine. Acetylcholine is one of many molecules known as a neurotransmitter. Since nerve fibers do not touch the muscle, the neurotransmitter must somehow carry the message across the synaptic cleft to the muscle fiber. Adjacent to the synaptic cleft, the sarcolemma greatly invaginates and a high concentration of protein sodium gates are present. We refer to this region of the sarcolemma as the motor end plate. As you certainly recall, the plasma membrane presents with specialized integral proteins that mediate membrane permeability. The neurotransmitter acetylcholine binds to these integral proteins known as sodium gates. When acetylcholine binds, the protein changes its shape and opens a gate for sodium to diffuse into the cell. Sodium therefore rushes into the cell in large amounts. It should also be noted that potassium is able to move through this gate, and therefore, due to the concentration gradient, potassium leaves the cell, but at a much slower rate than the entry of sodium. The end result is that the intracellular side of the membrane at the motor end plate becomes more and more positively charged. In other words, it becomes depolarized. When the positive charge at the motor end plate reaches a threshold level, sodium gates open up all along the sarcolemma in an event known as action potential, and muscle contraction has begun.